people think about AI. They think about things like ChatGPT and other large language models. But when we use AI for drug development, we're actually learning on real world data sets, not large language models. So Jack, maybe could you tell us a little bit about the difference between using AI for daily activities or in the digital world as compared to AI for drug development?
Yes, Shoshana, great question. I'll start off, and I'd love for Nadia also to add in information about the SARE data set, other data sets that we produce. But you're exactly correct, Shoshana. If you want to really impact the real world, right? If you want to say, hey, I'm not here to create another document, an image, a video. I actually want to develop a new molecule that's going to interface with some target in the body. And so let's say we're going after a certain cancer. We might be going after Alzheimer's, Parkinson's. We might be in the cardiac space. These are all very important indications. And traditionally, it's been really hit and miss, mostly miss, when it comes to traditional drug discovery and development. In fact, the numbers, I think, as many of our viewers know, are quite sobering. It takes generally 10 plus years to develop a drug candidate. And when it gets to clinical trial, 85% failure in clinical trial. And almost more sobering, if that's not already bracing, 40% of that failure is in phase 3. The most expensive phase, Shoshana, as you know well from your deep background in biotech as well. So when we looked at this issue of how do we really come to the table with new tools, we can't use a model that's trained on words, on social media posts, on cat pictures. That is not going to be the right tool for the job when it comes to something like as serious as a cancer-fighting drug or diagnostic. And now what we need to do is have new data sets that did not exist before. This is not data you can download from the internet. This is not data you can just buy off the shelf. You've got to do real-world experiments and take that data and then supplement that with new equation-based data. People sometimes call that synthetic data, but I like to call it equation-based data to really make it clear where that data comes from. It comes from the laws of physics and chemistry. And we know those laws and we can augment these data sets. These data sets in turn can create new AI models, not the kind of ChatGPTs that you're used to of typing a prompt in. Those are not really fit for purpose here, but novel architectures that are very well suited. Knowledge graphs, neural network models, and others that are really well suited. But I want to hand it off to Nadia to talk about specifically the kind of data we've been able to produce.
Yeah, I want to touch a little bit on the novel architecture. So if we think about the evolution of a GPU, right? So we borrow from our friends in video games. Those are graphics, right? This is the birth of the GPUs. When we think about the LLMs, that's a transformer model that predicts the next one, right? This is a 2D sort of like linear model. Now, when we get into images, right, generative images, now you get into like multi-matrix multiplication models, right? So we're looking at generating from a two-dimensional image, a little bit more complicated. So the GPUs evolved. Now the GPUs have evolved even more so where we're modifying the firmware of the thing that controls the GPUs to handle what's called triangular calculations, where we're looking at 3 objects in space across a matrix. That is highly complicated. This is the type of thing that people thought we had to wait for quantum computers. But it turns out we don't, right? And so through our partnership with NVIDIA, we've been able to exploit this kind of architecture through our models. And that is the power of the LPM. You cannot compare what is a two-dimensional fragment of letters to the complexity of a molecule that binds to its target in three-dimensional space, taking into account everything that's happening around it. And so that is what we've done. And over the last couple of months, we've released to the scientific community numerous models, including SARE, which has the largest binding affinity set of co-folded structures. There's a molecule, a drug-like molecule with its target that what a person can use to start drug discovery, right? And so over time, we've relaxed the need to have the structures that you would have to go to a cryo-EM to get as an input to start drug discovery using computers. We've relaxed the need for that.
And just to build on what you said, AQ Affinity, which Nadia and team just released in the last 10 days to the world, public, open source, Shoshana, so that researchers can use it, drug developers can use it. AQ Affinity represents the frontier, the edge now of what is possible, both in terms of determining the structure of that new novel candidate molecule and the receptor. Could be a protein as an example. And also the affinity, the binding affinity of how it will actually attach. So this is a very exciting moment. Again, large quantitative models, LQMs, versus and complementary to large language models.
Yeah.